Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Wendt, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Randy Jirtle. I've actually been lucky enough to meet Dr. Jirtle in person at a PLMI conference, or Preventative Medicine Lifestyle Institute conference, run by Dr. Jeffrey Bland back in 2022 in Seattle, Washington. A quick introduction to my guest before we get started. Dr. Randy Jirtle joined the Duke University Department of Radiology in 1977 and headed the Epigenetics and Imprinting Laboratory until 2012. He is now a professor of epigenetics in the Department of Biological Sciences at North Carolina State University. His research interests are in epigenetics, genomic imprinting, and the fetal origins of disease susceptibility. Don't worry if any of those sound foreign to you. That is exactly what we are going to be focusing on today in our conversation. He is known for his groundbreaking studies linking environmental exposures early in life to the development of adult diseases through changes in the epigenome, and for determining the evolutionary origin of genomic imprinting in mammals and the human imprintome itself. In July of 2022, more recently, Dr. Jirtle and his researchers from North Carolina State published the full list of ICRs, or imprintome control regions. This represents the first time in history that these imprint regulatory regions of the genome were fully identified in humans. This is groundbreaking research here, everyone. So we are very lucky enough to be working with him on the Imprintome project. In October of 2022, with all of these human positions identified, my company, True Diagnostic in NC State, collaborated to create a custom methylation infinium microarray that can examine the entirety of the human imprintome. Furthermore, True Diagnostics signed an exclusive license agreement to use and develop this microarray and further imprintome research. We are now working with researchers worldwide to generate epigenetic health data using this new exciting tool. Dr. Randy Jirtle thinks this represents a major step forward in our ability to predict disease risk. He even stated in a press release with True Diagnostic that the identification of the human imprintome will allow scientists to determine, simply by sampling DNA from the blood or other accessible tissues, the role that imprinted genes, epigenetically altered by environmental exposures, play in the formation of all diseases and behavioral disorders. So please tune into this podcast. The first half of it will be reviewing that agouti mouse trial that he is extremely famous for, and the second half of it will be focusing on genomic imprinting. What does it mean? Why does it matter? And how can we move forward with this research? In today's episode, we talk with Dr. Randy Jirtle. Welcome to the Everything Epigenetics podcast, Dr. Jirtle. I'm excited to have you today.
Thank you for inviting me.
Yeah, absolutely. So I'm not going to waste our listeners' time. I'm really going to hop right into it. You are extremely famous and known for your 2003 seminal agouti mouse study. You know, it's not only been talked about or described as one of the most cited papers in genetics, but in science. So I really want to focus on that for a large portion of our interview today. Can you just tell us about this experiment, introduce the listeners, give us, the idea of influence from the environment on phenotypic expression. Let's really focus in on that study.
Yeah, I mean, it's interesting because if you do 2003 and 2023, this is the 20th anniversary.
Yes, I didn't even realize that.
Yeah,
well,
so you should
no better way to celebrate.
Huh?
Yeah.
I think I might actually write a paper on this because it would be interesting, I think, to people. Not just your podcast will cover a lot of it, but when you sit and read it, it's a little different. You can chew on it and things like that, which you can't do as well when you're talking. And it
Sure.
doesn't seem like it was that long ago. Obviously, life doesn't seem like that either, right? But it seems like it happened not very long ago at all, but yet it is 20 years ago that study was done. And just
Yeah.
to put that in perspective, that study was done before the human genome was published. So there was no genomic information known either about humans or about mice. We were really in the dark when it came to genomic structure at that time. And that makes a big difference on when you go back and listen to and read old papers, because people now can't understand how hard it was when you didn't have that information readily available. Geneimprint.com, which is our website, and we started that. My son actually started that when he was like 10 or 12 years old in 1995. I mean, it's a long time ago. And if you look at websites, that was like when nothing was going on and you still had dial-up. And that was established originally to bring to scientists, to let them know mutations that were occurring in the gene of the IGF-2 receptor. And that's how I got into the whole field of epigenetics, because there were no mutations, there were no SNP databases, none of that was available. It's hard to believe that now, given all the information that's available on the web.
Right. You can download your raw genetic data into a plethora of platforms, right? I could think of at least 10 off the top of my head. So it's, really widely available now. So it's crazy to think about all of the progress that we've made and how you've had such a profound impact on that.
Yeah, and the impact of this paper wasn't from the standpoint of information like we're talking about data. It was a conceptual change. It totally changed the way people viewed disease susceptibility. We didn't start this. Rob Waterland was the postdoc in the lab and did these experiments in my laboratory. But there was a little history before that. I mean, originally we weren't planning to use the agouti mouse. When he first came into, and you want to talk about this later on, when he first came into the laboratory, and in fact, almost everybody that was working in the field of epigenetics at that time was working in the field of what's still called genomic imprinting. And we'll talk about that later on. But that's how we got into it, because the IGF-2 receptor was the very first gene to be identified as being genomically imprinted by Denise Barlow. And right at that same time, we had the first evidence that that gene was functioning as a tumor suppressor gene. And in 1995, we provided the first genetic information that indeed that was the case. And when that paper was published in Nature Genetics, Denise emailed me and she said she always thought that this gene might function as a tumor suppressor, but had no evidence that that indeed was the case. Unfortunately, Denise died. Otherwise, that would have been in 2017. That would have been someone else that you would have definitely wanted to interview, because she was an amazing scientist, and she totally changed my life, because it's the reason why we got into the field of epigenetics. You need to say, well, how can these two things coming together get somebody? The other thing is that people don't maybe know. I started my scientific career back in 1970 in the field, even before that, in nuclear engineering and radiation biology. So I have no formal training in epigenetics. I bungled into this field, and I really have very little formal training, hardly any really, in the field of genetics. Most of my training was really in computer science and physics and math. So I come at it from a more quantitative aspect than I think many people. And I think that's why we'll get to this later, why the phenomena of the imprintome appealed to me so much, because it's an engineering, computer-based approach to doing science. And I like where we are right now, because we have a lot of data that we can analyze. And in the old days, that was not the case. So getting back to this here, we were just getting out and had just defined when the phenomena of genomic imprinting evolved. So here you have a radiation biologist, nuclear engineering, providing the very first evidence of when a phenomena of imprinting evolved. And we found that it evolved in mammals in a common ancestor that gave rise to what's called therian mammals, which are marsupials, and the eutherians, of which most mammals on Earth are a member of. So we got into the field of epigenetics through this evolutionary biology approach to determining when the phenomena of imprinting evolved. And you'd say, well, why would you get into it through such a crazy angle? It's because there were only two genes that were known to be imprinted, IGF-2 at that point and the IGF-2 receptor. And there were a number of really, really strong groups of people, primarily in England and in Europe, working on those two genes from every angle that you could think of at that point. So getting into the field of epigenetics or even genomic imprinting when you had no background in it was not easy. So we chose this because we felt it was probably the least likely that anybody would really be interested in doing this type of work. So that's how we got into the field of epigenetics at that point through the evolutionary biology work. It's interesting from my perspective, too, because I did have to take a biology course when I was in high school, and biology at that point was primarily evolutionary biology, and I really did enjoy it at all, because all you were doing is memorizing things, and I said, I'm never going to need to know this anyway, so I didn't learn it very well. So I get to tell people that are listening to this that are young, you never know where science and life is going to take you. So here we are now in this field, and we know about imprinting, and we have a tumor suppressor in which one copy of that gene is turned off naturally by Mother Nature. And it's the tumor suppressor, and you ask, why would something like this crazy stuff, why would that have ever evolved? It makes no biological sense. To me, anyway, it makes no logical sense. It probably makes a biological sense, otherwise it wouldn't have evolved, but it makes no sense to me. And I said, this has got to be really important. And I decided then that we were going to bring our whole field, our whole laboratory into the field of genomic imprinting and epigenetics. We got into it, as I said, through evolutionary biology. So the next question was, we knew already because of Denise Barlow's work that there's the imprint regulatory elements were what was controlling this funny level of expression. So the question we had was, is it possible that an environmental, because I've always had sort of a toxicology sort of background and angle to my research, even in cancer biology, is it possible that exposures to toxicological agents like non-genotoxic agents could potentially alter the expression? So now we've got a question, just a fundamental question, could alter it through altering the methylations of this imprint regulatory elements. And that's when Bob Waterlin approached me to do a postdoctoral fellowship in the laboratory. And we were thinking about this originally to look at the role. He was a nutritionist, so that's how nutrition got into this study. There's not a lot of big logic on here. It's just the way you came in, from what angle you were coming in on this thing. And then the more we talked, the more we realized that even if we saw a 10 or 15% change in methylation levels at these imprint control regions, that the primarily geneticists would say, well, that's not biologically relevant.
Sure. Yeah, absolutely. Yeah, there's I remember discussing this and hearing your story a little bit when I first met you at PLMI at Dr. Jeffrey Bland's conference in Seattle last year. So it's interesting to understand the background of everyone who is involved on this type of study. You all brought unique things to the table, right? Your specialties in each different area, and you come up with this miraculous study, what I would call it, right?
Well, it's sort of just jammed together, kind of. There were logical questions, but you're right. I mean, what the angles we came at it from were very different. So then both of us had read these papers about the agouti mouse, and the beauty of the agouti mouse is that it had already been known that epigenetics was involved in controlling coat color, and as a consequence, also that was associated with disease susceptibilities of obesity and diabetes and cancer.
Is that why you chose that model, the agouti mouse model?
That's absolutely correct.
Perfect.
Now, I want to make it clear that the agouti mouse model is another class of epigenetically regulated genes that occurs very, very early in development. They're not those genes that are expressed that way are called metastable epialleles, and that's another set of what I call sort of epigenetic methylation dependent regulatory elements for genes. There's two major classes. One's the metastable epialleles, and one other classes of genes that are genomically imprinted. And there's a lot of other methylations that occur for differentiation and stuff like this, but the ones that are very, very early change that are involved in disease susceptibility are those two major classes. And Rob is now working on the metastable epialleles, and he's defined the metastable epialleles in humans. That's been done, and it was done before we were able to define the imprintome in humans for imprint gene regulation.
Right.
But now both of these groups are defined.
Yeah. So can you define both of those groups just to make sure our listeners understand as you're walking through this? And because you've mentioned
to explain the epigenetics, the agouti mouse study without looking at these animals. So you've got coat color. Let's just focus on coat color, but also disease susceptibility is dependent upon methylation of a transposable element that's upstream of that gene. And this is a weird strain of animals. It's not true in the normal mouse. This is a mutant, basically, but a very, very effective mutant. And it provides a wonderful model system for addressing clearly as you can in biology, what role, for example, in this situation, nutrition and putting methyl groups in the diet, which is like folic acid, betaine, I can't remember. There's a couple of other groups of nutrients that are involved in providing methyl groups. All the methyl groups that are then put down on the DNA and the histones come in from our diet. So that's why we thought if we loaded the hopper, in other words, fed the mother lots and lots of methyl groups, we might be able to shift the coat color to brown, which is the healthy animals, and has lower disease susceptibility and et cetera, away from the yellow animals. And we could then demonstrate or test whether or not that was through changes in the epigenome itself. And that's what our study did for the very first time. And as a consequence, it really started what I call the field of environmental epigenomics. It started with the publication of that paper. There were a lot of people that were working on this, but they were primarily biochemists, so they were in the signal transduction pathways, but that wasn't the memory system. The memory system is putting the marks onto the DNA and histones and turning genes off and opening other genes up. And that's how exposures early in development can cause or actually reduce the incidence of diseases in adulthood. So this then was and is the mechanism for the fetal origins of adult disease susceptibility. And that's why this paper is so fundamentally important, because it provided a mechanism for something that made no sense. And up until that paper was published, in reality, a lot of people just didn't believe it, didn't talk about it, and swept it under the rug. They don't do that anymore. They might still be arguing about different aspects. But I think that point has now been made.
It's something that can no longer be ignored. And I really want to point something out there when you talked about shifting methylation markers. I actually heard someone else say that I talked to, but that's a great point, because we want to shift methylation markers in our favor. A lot of healthcare providers that I even talk to now, when we talk about DNA methylation or epigenetics, they still think about the MTHFR gene and the COMT gene. They always think that this methylation is a bad thing, right? But we're trying to teach, if you're giving those methyl donors, in the example with the agouti mouse trial, and you're shifting that coat color to be favorable, you're going to get that better outcome. So I love that study. I just think it's groundbreaking. It's something that, you're right, people can't ignore now, right? They have to deal with it.
You would hope so, but I mean, even still, I mean, it takes a long time. I mean, 20 years is quite a long time, but I think it's starting to become clear to a lot of people. And my analogy is, again, because of coming through computer science and engineering, I think of the DNA as being comparable to the hardware of your computer, and the epigenome is just simply the software that tells that computer when, where, and how to work. And this is how you can get basically 260 or so different cell types in your body from a single cell that had all cells have the same DNA, but every cell in our body, I mean, you look at us, we're everything is different. And the reason for this is because the programs running in those cells is different, and they were established very, very early. That's in during development. And that's why that stage is so critical for maintaining good health, and also alternatively messing up your health later on in adulthood.
Yeah. And I actually used that example because I remember you said that at the conference and I loved it because, I usually say, your eyes and your heart, they have the same genetic information, they have the same DNA, but what makes your eyes and your heart? And it's those epigenetic mechanisms, those on and off switches that are going through the central dogma, or maybe not, and then resulting in a phenotypic outcome, or again, maybe not. So I really love the software and hardware example that you gave. So rounding the end of the conversation about the agouti trial and the mouse study, did you know you were going to have such a profound impact on the scientific community? Or, I know you talked about your background, but did you know you were ever going to reach this level if, you would call it success?
Well,
I definitely would.
I told Rob, I said, if this experiment succeeds, and by that I meant in this situation that it went according to our hypothesis. Now, I must say that many times in the field of epigenetics that what I hypothesize is not correct. There have been a mini paper that I've already written in my head that ended up not being supported by the data once they're out. But in this situation, it did end up the way we thought. But I told Rob, I said, either we're going to be going down in flames or we're going to probably be famous for this because of what it does fundamentally in providing a fundamental mechanism for the fetal origins of adult disease susceptibility. And what it also does is scientists don't believe things very much. They like to be, they're the doubting Thomases of science, right? They want to be shown that this is correct. And that's what our job is to be doubting on everything. And before there was a mechanism for this, the phenomena of the fetal origins of adult disease susceptibility, which was really mainly introduced by Barker, it was even called the Barker hypothesis back in the old days. There were people that believed, but a lot more people that just didn't believe it or just didn't even think about it. You can't do that anymore. I mean, I don't think most people can realize that you can't fix a computer unless you're both looking at the software and the hardware. And it's the same thing for disease susceptibility, period.
Yeah, absolutely. And again, you've given us this great research now that we can build on top of. So, to summarize there, your first experiment showed there's this environmental and even nutritional exposures that can determine your phenotypes through epigenetic regulation and other mechanisms. This means our diet, our exercise, our lifestyle, all of these triggers, our food, the exposome, the toxics that we're exposed to, and even our mindset can actually impact our health outcomes. So
the other thing I was going to say is that in a additional study later on with Dana Dolinoy, she demonstrated that the negative effects, for example, that we showed with bisphenol A, there we shifted the coat color towards yellow, could be completely negated by also supplementing the mother's diet with methyl groups. So in effect, your diet was also potentially can reverse possibly or negate the negative effects of things that you're being exposed to in the environment also. So it's a yin and yang. It's moving back and forth, and it's plastic.
Yeah, there's this change occurring. So rounding all of that together, right? And this is why I wanted to have you on and how we're working with you now, which we'll get into. But that established the idea of the imprintome that you hinted at the beginning, right? So
keep going, Anna.
Yeah, no, we're just going to have you explain what imprinting is. What's the imprintome? Give our listeners an insight and be as detailed and specific as possible because this is still such a new idea, right? So feel free to go on a whim here.
Well, I mean, I thought about this back when we were doing the original studies and the original evolutionary biology studies. In fact, I put a grant application in to determine the human imprintome back in around 2005 or so to the NIH, and it was obviously never was funded. And I'm going to make this point to students and stuff too. Everything that we are actually known for was not originally actually supported by NIH. Ultimately, NIH supported things that came from this information, but not the original concepts. I mean, there's no way I would support myself to do an evolutionary biology study, given my background. But yet,
right.
Yeah, and it's the same thing with the agouti mouse. I mean, it's hard to believe very many people would have supported it at all, because a lot of people didn't believe it. So it doesn't. Those kinds of changes then have to be supported by other kinds of money, more hardcore money, private money usually, and company money in some respects. And that brings up the agouti mouse. So we had enough money set aside that we could do the experiments that we did. So we didn't need NIH to support that original study. The other ones were all supported by NIH, and they were great studies too and had great concepts, but the original one. And Rob Waterland came in here, and he was supported for 2 years as a postdoc by a grant from Dan & Yogurt. So I have a soft spot in my heart for Dan & Yogurt.
They helped you out.
They helped me out when we needed it. So we got that done. So even at that point, we were thinking about these imprint regulatory elements and the fact that we needed to know those. And it was from our evolutionary biology experiments and also other people's experiments. It was becoming very clear that different animals had different repertoires of imprinted genes. So you can't go to the mouse and say, well, I'm going to do all these studies in the mouse, and then I will know the repertoire and where all the imprint regulatory elements are for the human. It doesn't work that way. We're different. And in fact, it's possible that once the phenomena of genomic imprinting evolved, that eutherian mammals basically evolved themselves on the backs of the development and formation of imprinted genes themselves. So they could potentially, and I do think this is correct, but I'm not sure how you would test it, that was a driving force in mammals for evolution, rapid evolution, because you're doing it on the back not only of mutational changes, but also now a phenomena that you can change the expression dramatically in time and space and sex. And if you think about that, that's incredibly powerful. So we needed to know the imprint regulatory elements in humans. And there was a lot of different diseases already at that point. Beckwith-Wiedemann syndrome, Prader-Willi syndrome, Angelman syndrome, on and on and on. There's a number of syndromes that were known to be created in developmental disorders and cognitive disorders that were being affected by inappropriate expression of imprinted genes.
Yeah, definitely. No, I love it. And we're going to get into those disease states as well.
That's where we were, Hannah.
yeah, that's where you were. And
that's where we were in about 2007. I first defined then what the imprintome was in the first editorial for a journal called Epigenomics, which came out in 2007. What the imprintome was and why we needed to define it. But telling you why and doing it are two different things.
Definitely.
Now I'm going to let you ask another question, and then we'll get into what imprinting is and stuff. Anything you want?
Well, I just wanted to add there, my definition. I want to see if what I'm saying is correct. So for most genes, we have two working copies, right? One from mom, one from dad. But with imprinted genes, we only inherit one working copy. So depending on the actual gene itself, either the copy from the mom or the copy from the dad is epigenetically silenced, right? If something is methylated, it's turned off, it's not being expressed. So that is exactly what we're talking about. Silencing usually happens through the addition of those little tiny methyl groups during egg or sperm formulation. So these, you can call them epigenetic tags on imprinted genes, usually stay put for the life of the organism. And that is really, really important. But
and they're established extremely early in the gametes or right after fertilization.
Exactly. And they are reset during the egg and sperm formation, right? So regardless of whether they came from mom or dad, certain genes are always silenced in the egg, and there's always certain ones that are silenced in the sperm. So, I think this is correct. Humans have about 20,000 to 25,000 genes, and imprinting is unique to mammals, right? That's where you started to do a lot of that work that you just mentioned in
Let me add one other
your
thing,
biology
Hannah.
classes. Yeah,
They're not. It's not actually correct to say they're unique to mammals. In animals, they're unique to therian mammals, but there's one other class of living things on Earth that also has imprinted genes, and you know what it is.
Flowering plants.
Exactly.
Is that correct?
But it evolved independently. So there's
Yep.
two classes, basically, of living things that have imprinted genes.
Yep. And
Yep.
and is it correct to say in mammals only about 1% of those genes are actually imprinted, right? It's a very, very small subset.
It depends on what you say. Like some people say there are 20,000 genes. Now I've read that, if you look at splicing differences and all that kind of stuff and RNA and things, are up to like 65,000 genes in the human. So it's probably somewhere's about 1 to 5%, I would say.
Okay.
Somewhere's in there, but it's probably more dependent now about how many genes we have rather than than the ICRs, because I think we pretty accurately define that number, and that's right around 1,500. Somewhere's in that range.
Correct. Yeah, I was, doing some research before I hopped on with you, and I know you just identified right the entire list of those imprintome control regions. I have 1,488. So yes, 1,500 of them. So how did you identify those imprintome control regions? I know you just published that with NC State, and that's how True Diagnostic, my company, and we're working with you. How did you actually identify those and publish about that?
Well, the first thing we had to do was get support to do it.
Yep.
And as I said, I failed when I tried, and it was around 2005. I had another shot of it, because all the work we were talking about before, I was on the faculty at Duke for 35 years. So the vast majority of work and research that I've done, it was done at Duke University. And before that, it was at the University of Wisconsin. So that's how I got into radiation biology from nuclear engineering. But when I got to Duke, was doing cancer research, because that was sort of my background as a postdoc. And that's how I said how we got into the phenomena of epigenetics. But when I retired, I thought I was retired. But Catherine Hoyo and a number of my people moved over to North Carolina State University to set up an epigenetic and epidemiological group over there in humans. And Catherine asked me if I'd be willing to help do that. So I am now an adjunct professor at NC State, which is a lot easier for me because I don't have to take care of the day-to-day runnings of things and stuff like this, but I'm still involved in, writing grants, for writing papers, all the things that, not so much getting in the lab anymore, and not even getting in the lab every day kind of say, but now with Zoom, I can do that a little bit better because Durham, where I live, is about 30 miles from Raleigh. So I get over there, but not as much as I had to and did do, and I love to do when I was at Duke, because I only lived 3 miles from my lab. So I kind of like lived in my laboratory, except when I was home with the family.
Yeah.
But that's different now. So the whole thing then, the first thing is, even though you have a great idea, is getting support. And that was not easy. And that was really done by Catherine Hoyo. And she did that based upon the fact that we knew that imprinted genes were playing a big role in diseases and in the environmental responses in disease formation, because that's the type of epidemiological research he was doing. And the support to do the human imprintome then came through NIH in this case, actually, to do that. So about 3-4 years ago, we got the support to do it. So now the question that you had is, what do you do now that you've got money to do this? And when I first thought about doing this in 2005, we could have pulled this off, but the sequencing ability was so rudimentary compared to what it is now, that it would probably have cost 10 to 100-fold more money and taken a lot longer to do than we were able to ultimately do with the sequencing ability that we have now. a lot of science really does depend upon, it's not just sequencing, as you know, because you've got all these little pieces and you're looking for literally needles in the haystack is what you're looking for now. So it's the computer facilities. I could not have done this project at Duke. You say, why? Because Duke does not have the computer capacity and the engineering bioinformatics capacity and the storage capacity that was needed to put all these little pieces together and to come out with the 1,488 piece little regions, which there are only hundreds of bases, some of them thousands, but tens of bases to hundreds of bases. They're small little areas. Without those facilities, because NC State is an engineering school. And they didn't set up all these facilities to be nice to biologists. They primarily probably did this because of physics, because they generate a lot more data even than what we're able to generate with sequencing. But because we're at that facility now and at that university, we were able to use their incredible facilities and computers and storage and bioinformatics people to pull this off. So what does pulling this off mean? You have to look at, you already said this, these imprint regulatory elements are, once they're set, so at that imprint regulatory element, one copy of the genome will be unmethylated, it'll be like this, and one will be methylated. So when you sequence that region, it'll look like that little area is 50% methylated. Now that's oxymoronic if you think about it, because there's no such thing as 50% methylation, but what it means is one copy is shut off and the other one is functional. That now gives you, at that little area, 50% methylation. So you're looking through the whole 3 billion, or is it 3 billion bases for areas where you have 50% methylation.
Really a needle in the haystack.
But when
if I could,
Anna, when you look at that website and look at those genes, it damn near brings tears to your eyes. They are 50% methylated.
Yeah, I know. And your website is such a great resource for anyone listening. I'm definitely going to put it in the show notes. If I could go back and redo schooling or recommend, if I have children one day, I'm going to tell them, I would, hope or push them a little bit towards science first off, but put them toward the bioinformatics or, the computer programming and all the artificial intelligence models. We just can't function without that. Like you said, NC State has such a great facility, and without that, we wouldn't have even been able to identify these. I think that's super important as well to know.
Yeah, so the other thing that I knew, and you knew too, is since these are established in the gametes or right after fertilization, that means in the embryonic stem cell stage of development, there are no differentiated cells at that point.
Yeah, that was one of my questions too. If you kind of summed it up there, but if you want to explain the difference between the somatic and the germline imprintome regions, right?
Well, they're the same regions, but they're methylated differently. So if you look at somatic cells, because these marks were established either in the germline, where one copy now in the germline, if you look in mother or father, sperm and egg in humans, now we have to be in humans, both, you will find that at these regions, one copy, either the sperm or the egg, will be 100% methylated in these regions, and the other one will be 0%. And you'll be able to show this clearly. Now, when they come together in the formation of after fertilization, one copy from mom and dad, in these areas, they show up as 50% methylated.
Correct.
So if you look down now where you have somatic cells, and that was the other thing we added because you had different cell types that we looked at rather than a single one. Why? Because it's got to be common and the same all over the body. If it's not an imprint regulatory element. It would be dependent upon differentiation, that kind of stuff. We don't want those. It's not that they're not important, but they're not imprint regulatory elements. So the whole trick was to find regions that were consistent across all cell types in the body, but were different one methylated, unmethylated in the germline. And when you see that, it darn near brings tears to your eyes, because that is the imprintome. That is the imprint regulatory element. And you can see it as clear as day.
Right, right. I know, so groundbreaking, and again, something that I'm just really excited to work with you and NC State on, and Dr. Hoyo. You know, I definitely want her to have her on my podcast. I need to reach out to her.
Yeah, she'll have a much more epidemiological spin to this whole thing, but I think hopefully she'll explain that the importance of epidemiology in getting this funded.
Yeah, because I know that's her background. I know that's her specialty. So the entire reason, well, I would say one of the reasons we did this is to obviously make it, you know, for researchers to be able to study this and look into it a little bit more. So the imprintome control regions and just the imprintome itself, they're critical spots for development and growth. They play a major role in developing diseases and disorders, you know, obesity, Alzheimer's, autism, bipolar disorder. I mean, we could name a lot, I'm sure. So, you know, schizophrenia even. So tell us how we can connect the imprintome to those different diseases. How are we starting to connect the dots here?
Well, the theoretical reason why imprinting evolved, because if you think about it, every one of these genes now are disease susceptibility loci.
Yep.
The reason for it is because you only have a single functional copy, and they're literally all growth regulatory. Either positive, and in the tumor cancer world, that would be called an oncogene, or they're inhibitory, and in the cancer, that would be called a tumor suppressor gene. So my guess, and it's just a guess, that there is no cancer, none, that doesn't have dysregulated imprinting, potentially as some of the earliest changes in the development of the disorder. Why? Because these protect, when you dysregulate them, they protect the cell from dying. They're able to continue growing. So you inactivate a tumor suppressor gene. So now it continues to live. You inactivate the apoptosis signaling pathways. Many of these genes are involved in that phenomenon. And when that happens, the cell can continue to accumulate oncogenic events without dying, and ultimately shows up as a lump and a bump that we call cancer.
Sure.
So that is, to me, is almost a given. But interestingly, because they evolved to control growth, because the reason why people think that they evolved in the first place was to control the amount of nutrition that the offspring extracts from the mother, with the father trying to maximize that extraction and the mother trying to tamp it down. And this is all basically, so it's literally a battle between the sexes to propagate your genetic information forward. That's the theory. Now, if indeed that's the case, you can see why growth regulation is so very, very important, because you're really controlling the growth at the earliest stage to try to give your offspring the greatest opportunity and ability to grow and survive in the future. And then to propagate the genes forward into the next generation.
Sure, yeah. And you know, we also saw Dr. Michael Skinner from Washington speak about that, right? The translational or transgenerational epigenetics. And that's a whole other world. You know, I want him on my podcast too. So it's all intertwined and work together.
Yeah, definitely. And some of the genes there that probably are being altered, that are giving maybe rise even to transgenerational effects, might be imprinted. I don't know, because that's something that can now be looked at.
Right. Yeah. Now, because you've discovered it all.
Because you can screen. You don't have to look at 3 billion bases. You're down to 22,000 bases. 22,000 CPG sites is all you need to screen now. So it's a much easier task. And as you know, we're in doing this. The first attempt to do this is to make a chip that will, a DNA methylation chip that will allow for relatively easy screening of these various ICRs in any kind of disease there is, and determine whether or not imprinted genes are deregulated. And my guess is that there are going to be a lot of them.
Right. Yeah. And especially in those disease types that we just named.
Right.
So, again, wrapping this up, there's about 1,500 imprintome control regions and about 22,000 CPGs. So that's really how Dr. Jirtle and I started working together, is because my company, True Diagnostic in NC State, we're creating this, what we call, I'm pretty sure we're calling it the Imprintome array, where you can actually quantify the methylation at those positions. So now you're able to measure this, and that just unlocks, hundreds and hundreds, thousands, millions of doors into these different disease pathways. And the, really the sky's the limit now that we can measure these. So I'm super excited to see what people do with this and what comes of it.
Well, it's hard for people to put their heads around it. And when I wrote up, I don't know where it was, might have been in a review or another review that I had, I think one that was published, but it could be verbal too. But it's not an overstatement. What you can do now, once you have the imprintome, is you can determine the role of genomic imprinting and its deregulation epigenetically, and really probably even ultimately even genetically, but for sure epigenetically, for every disease and disorder that we have.
Yeah.
Now that doesn't mean that those genes are involved in all of those, but that can be done relatively easily. And my guess is that many of the major, and including behavioral disorders, you were talking about autism and schizophrenia. If people that are listening to this are interested in why those two diseases or disorders are so interesting, I would recommend they read the Christopher Badcock's book called The Imprinted Brain. It really outlines why that is probably, these two disorders are diametrically opposed and due to different imprinted genes being deregulated. In one case, in autism, potentially an overexpression of the paternally expressed genes in the brain in early development, and then it continues on to give the phenomena of autism. And in the other case, an sort of an overexpression of the inhibitory genes, which tend to be more maternally controlled. And that gives rise to potentially schizophrenia. That theory now, with the imprintome being defined, can actually be for the first time tested. Now, whether someone is going to get the resources together to do this, and that doesn't mean just monetary resources. It means getting the samples together to do this. I'm not sure, but I'm sure at some point this will be done.
Yeah, I don't doubt it.
Hannah, it's of fundamental importance to know this.
It is. Yeah, I couldn't agree with you more on that. And that is something that we'll just have to, again, hope and wait and see. I know there's a lot of, biobanks out there now with those disease samples in states. So hopefully those resources, after they have the monetary resource, they can move on and find a really great bank with some samples. So, Dr. Jirtle, probably one of my last questions for you, we'll see where this takes us. What are you excited about? You have all this work, you have this great background, you're, again, one of the most cited papers in history and in the scientific community. What are you looking forward to? What do you want to understand and know?
If I could live for a longer time.
Yes.
You're asking me what would I address right now?
Yes. What excites you?
I would work on the potential role of genomic imprinting in human evolution.
Okay. Yeah. Tying your past studies with what you know now and trying to dig a little bit deeper into
because
that.
What I feel is that we're going to find that when we start looking at that, what you have to do is look at different imprintomes now in different species and start determining which imprinted genes, imprint regulatory elements. You keep focusing on the imprint regulatory elements because that's the common thread through here. What genes these imprinted regulatory elements control, we don't know. So in that list, in that paper, we've just put the gene that was like it was right, the imprint regulatory element was right in the middle of the gene or it was very close to it. But it
Gotcha.
could be that gene, and it could be other genes that it controls, or even some of them, they're not actually true imprint regulatory elements, but a goodly number are going to be. But if you focus on the ICRs, imprint control regions, then you can compare one species to the other species, and you can start getting an idea how those regions changed as animals evolved from basically, we don't have a precursor to marsupials and eutherians, but you can get down into that region by going to the marsupials, and then you could even use platypus as your outgroup. They don't have imprinted genes, or the echidna, or chickens, which is what we use as our outgroup. And follow that. Again, you need great bioinformatics and great ability to store and manipulate tons and tons of data. So it appeals to me a lot from my engineering background.
Yeah, it's almost like a map you would be creating, right?
Exactly.
Between all of the different species and figuring out where are the connections, how are these intertwined. So I think
you can never prove anything, Hannah, like this, but you can start seeing trends. And this becomes ultimately the weight of the evidence suggests that this is something that was really critical. Let's say the addition of imprinted gene or how it was regulated is it not in the chimpanzee, or is there? And it's you know what I'm saying? And you can go back and forth, and you can start. So you say, well, what is this gene? What is it doing? So you're starting to get a better feel for, in effect, what made us uniquely human at the genetic and epigenetic level.
Yeah. Understanding patterns, connections. I think there's someone out there listening needs to study that. And it's all about, I could ask you hundreds of more questions. It's all about asking the right questions. And
the pragmatic ones are the ones we're working with True Diagnostics and other people are going to be the multitude of behavioral diseases and disorders. And my guess is ultimately cancers too. I mean, I really do believe that it is an important thing that individuals are very, very interested in, and it's going to be an important thing to dig out the role that imprinting plays, because it potentially provides an epigenetic mechanism by which you can treat.
Of course,
and it's an epigenetic mechanism by which you can diagnose.
Exactly, and identify early, and again stop that disease from maybe
even these markers are in the blood. I mean, I don't know, but I mean, that's where those are more what I would say immediate pragmatic things that are important to people that are here. But you asked me a different question. What would I do if I had unlimited money and unlimited time? I would work on the role that imprinting played in the evolution of mammals.
Yeah, still important, but understand the pragmatic answer as well. So it's like, you have the answer to one question, and then that opens or that causes you to have a ton of other questions. So it's like we keep learning and evolving ourselves. So I think it's super exciting. Well, Dr. Jirtle, I appreciate your time today. This is my final question to you. I think you will find it especially interesting. If you could be any animal in the world, what would you be and why? And I know you have your stuffed animals there behind you as well. So this I'm excited to hear your answer.
That's a weird question. That's giving me time to think about a weird answer.
Yeah.
I don't know. I mean, it sounds not very exciting, but I like what I am.
Really? Okay.
Yeah, because other animals can't do what I'm able to do and what I'm able to study and find with the technology and techniques that we have right now.
Very valid answer. No, your answer is valid.
To me, it's extremely, I'm not kidding, it's extremely exciting. I've never been in my research career at such an exciting time as right now. And the only thing I would like is make sure that I'm around long enough to do it.
There you go. Well, that just proves, how devoted you are to your work and uncovering more and just, making new findings and discoveries. So, no, Dr. Jirtle, I really appreciate your time. Where people can find you is your website, the geneimprint.com. Is that correct?
That's our website. Yes.
Okay. your website, and I'll put that, like I mentioned before, in the show notes for everyone. So, thank you for joining the Everything Epigenetics podcast. And remember, you have control over your epigenetics. So tune in next time to learn more. Thanks so much, Dr. Jirtle.
And that's what I said. I want to be still. I want to be a human.
No, answer is definitive. And that's very unique. I don't think we'll ever have anyone else say that same answer. So I'll have to keep a tally and come back to you.
Okay.
All right. Thanks, Dr. Jirtle.
Bye.